Hi everyone, today we'll discuss in more detail the materials and methods section of this paper, role of glutathione on acrylamide inhibition, transformation products and mechanism. This study was published by Zhu and colleagues on the Journal of Food Chemistry in May 2020. So here's an overview of the materials and methods section, but today we'll talk about two of the main steps highlighted in red: the characterization of intermediates, for which an ESI QTOF mass spectrometer was used, and the quantification of key compounds, in which an UPLC TQS mass spectrometer was used. So let's define these tough acronyms and understand them better. This is the instrument responsible for characterizing the intermediates in the reaction. The ESI, or electrospray ionization system, produces ions using an electrospray by applying high voltage to a liquid to create an aerosol. Very little fragmentation of molecules results from the ESI. Little structural information is obtained, reason why it is also paired with the QTOF MS in this experiment. The QTOF MS, or quadruple time-of-flight mass spectrometry, produces a spectra through a quadrupole, a collision cell, and a time-of-flight mass analyzer unit. A quadrupole consists on 4 parallel rods arranged in a square formation selecting for specific ions. The ions coming from the ESI source are accelerated down the flight tube to the detector, which determines the ions' mass-to-charge ratios. Lighter ions are actually faster. And additionally, this unit combines 2 different mass analyzers, which allows for efficient detection of mass-to-charge ratio of analytes, a desirable feature with a variety of analytes on this study. So this is one of the figures you'll find on the paper that relates to the ESI QTOF MS, and it shows the mass-to-charge ratio on the x-axis and percentages on the y-axis. On this example, 3 molecules were recognized, as you'll see on this slide. This figure is also obtained from the instrument, and here are highlighted the 3 analytes that were on the previous slides. DFGSH, which is an amidori product formed through the reaction of glucose and glutathione, and also AACYS and the AGSH, 2 byproducts of the reactions between acrylamide with glutathione and cysteine. So for the quantification of the compounds that have been identified in this step we just discussed, the authors utilized the equipment on the picture paired together. UPLC, or ultra performance liquid chromatography, follows the same principle as HPLC, or high performance liquid chromatography. HPLC consists in the separation of compounds on a stationary phase column. A mobile phase runs continuously through the system, pushing the sample through the column. In this study, the eluents were acetonitrile and diluted formic acid. An autosampler sends the sample to the column, which separates it, and finally to the detector, which receives the result of the sample separation. These results are translated in a chromatography data system, given a chromatographic spectrum that provides the quantitative and qualitative data about the sample. Peaks are plotted with respect to the retention time, and concentrations can be calculated. The UPLC has a few advantages to the HPLC, which derive from a difference on the column. The UPLC column has a mean particle size diameter smaller than 2 microns, which results in higher pressure in the system and optimal linear velocities. A TQMS, or tandem quadrupole mass spectrometer, differs from traditional MS techniques as it contains 2 quadrupole mass analyzers in series, with an RF quadrupole between them acting as a collision cell, thus allowing for mass analysis to occur in a sequential manner in different regions of the instrument. It is an easy to operate, highly efficient, and sensitive instrument. The ex vivo TQS offers 6 orders of magnitude of linear dynamic range, which is important for quantifying compounds in samples where the concentrations of individual analytes differ greatly. The TQF is a great instrument that quantifies the concentration of specific substance present in a sample, which was also its main purpose on the study. So this slide depicts figure number 1 in the study, where the authors find their hypothesis to be confirmed: glutathione inhibiting acrylamide. The concentrations are obtained from the quantification results in UPLC, TQMS over time. And finally, on figure 3, more graphs depict the concentration of various analytes, also obtained from the UPLC TQMS. Thanks for watching.